So in this video, we're going to continue to discuss the small intestine, and we will continue with the types of secretions that are either produced or received by the small intestine. So really, the major secretion of the small intestine is the intestinal juice. We make about 1 to 2 liters a day. And again, most of this intestinal juice is water, very much like we saw in the gastric juice. We're going to see mucus. And unlike gastric juice, intestinal juice has an alkaline pH. So looking at a pH of about 7.4 to 7.8. Besides making the intestinal juice, and that's important to distinguish, so the small intestine makes the juice, but in addition to that, the small intestine is also going to receive certain products. So among those products is number one, bile. So we know that bile is made by the hepatocytes of the liver. And it can be stored in the gallbladder. Either directly coming from the liver or coming from the gallbladder where it gets stored, this bile is going to enter the duodenum through the bile duct. Bile is extremely important for digestion of lipids. Because it has bile salts, lecithin, and they're extremely important for emulsification of fats. So basically, bile helps us break down lipids physically into smaller parts so that lipases can then act and start breaking them down enzymatically. The second product that the intestine receives, and that also happens in the duodenum, is pancreatic juice. So pancreatic juice is received from the pancreas, and it enters the duodenum through the so-called pancreatic duct. The importance of a pancreatic juice stems from the presence of multiple types of enzymes. So pancreatic juice is going to have a pancreatic amylase. We know that is important for breakdown of carbohydrates. It has pancreatic lipase, which is important for the breakdown of lipids. We have a pancreatic RNase and DNase for breakdown of nucleic acids. And finally, we are going to have pancreatic proteases. This pancreatic proteases actually mention some additional details. So I'm going to write them down one more time here, pancreatic proteases, because they are produced in the pancreas in form of zymogens. And if you remember, we talked about pepsinogen being a zymogen, which means an inactive enzyme. And that means they have to be activated. So pancreatic proteases are produced in the pancreas as zymogens. They're going to pass through the pancreatic duct into the duodenum, and in the duodenum they will be activated. So trypsin and chymotrypsin are the most important proteases of the pancreas that get activated in the intestine. And finally, the last thing that we're going to write here for secretions is brush border enzymes. So these brush border enzymes are really important for final breakdown of carbohydrates and proteins. So in order to absorb carbohydrates and proteins, they have to be broken down all the way to monosaccharides in case of carbohydrates and amino acids for proteins, and that is achieved by the brush border enzymes that are produced in the small intestine. Next thing we are going to mention here is motility. Remember in the alimentary canal, we have lots of peristaltic movements, so peristalsis. We know that peristalsis is dependent on the enteric nervous system, primarily the myenteric plexus, that part that's located in the muscularis externa. And we're going to talk about two types of movement here in the small intestine. The first one is called segmentation. Segmentation is a localized movement. And what segmentation kind of looks like is ring-like contractions of the muscularis externa. And so if we imagine that this is the small intestine, we're going to have ring-like contractions in different places of the small intestine. And what segmentation is doing is basically mixing the contents. Mixing intestinal content. Segmentation a lot of times is explained as a massaging action of the smooth muscle in the small intestine. So it mixes the intestinal content and it also increases contact with the mucosa. And that's important because small intestine is the primary place where absorption is going to happen. So we need to make sure that this contact time is increased. The frequency of these waves in the small intestine is slightly different. So in the duodenum, they're about 12 times a minute, and that decreases to about 8 times a minute in the ileum. The second important movement is called migrating motility or motor complex. So migrating motor, also referred to as motility complex. And the primary function of this migrating motor complex is transport. So we need to be able to move the contents of a small intestine from the duodenum all the way towards the end, towards the ileum, into the ileocecal valve, and then into the large intestine. So those are long waves. And the migrating motor complex is actually stimulated by motilin, which is produced by those M enteroendocrine cells. And finally, the last thing that we're going to discuss for the small intestine is functions. So we know that the digestive system has 6 functions, and let's see which functions are associated with the small intestine. The first one is obviously propulsion. We just talked about the migrating motor complex, so ability to move contents into the next part of the alimentary canal, so propulsion. We're doing some mechanical digestion. Think about the segmentation, so kind of massaging the contents of the small intestine and increasing the contact with the mucosa. We do a lot of chemical digestion. So think about all the enzymes that we see in pancreatic juice, the brush border enzymes, and then the help from the bile in terms of digestion of lipids. And finally, and one of the most important things that happen in the small intestine, so I'm gonna write it with slightly larger letters, that's absorption. So we talked about this large surface area of the small intestine, and that is crucial for making sure that we are moving nutrients either into the bloodstream or into the leg shells, if we're talking about lipids, and we can then use those nutrients to power up different activities in the body.